Cells in a normal animal are maintained in a healthy and functional state by regulation mechanisms. The normal cells die after a limited lifespan and are replaced by new cells. Due to various causes, regulation mechanisms lose control over certain self cells of the body. These cells divide indefinitely and form a mass of cells, forming clones and give rise to a tumor or neoplasm. Cancer and tumor. Cancer is a group of diseases which are characterized by uncontrolled growth of any of the self cells of the body. Cancer cells can also be called as altered self cells which have escaped the growth control mechanism. Tumor is used as synonymous to cancer sometimes, but tumor basically refers to a mass of cells which can be termed as benign or malignant. Increase in number of dividing cells or increase in cell longevity or divisional rate. Normal cells inside the body get transformed to malignant cells due to various reasons. Transformation is a process in which the characteristics of a normal cell changes and the resulting cell is known as a transformed or malignant cell. Transformation may occur in cells due to physical carcinogens such as ultraviolet light and ionizing radiations, chemical carcinogens such as DNA alkylating reagents, viruses, for example, SV40 and polyoma viruses. They can also induce malignant transformation in the cells. The transformed cells may be characterized by decreased requirements for growth factors and serum, anchorage independent, growth in a density independent fashion, immortalization. Tumors developing in the tissues due to above transformation may be benign or malignant. The tumor developed by cells which divide indefinitely at one site in any tissue or organ to form a mass of cells, but do not invade any other healthy tissue to develop another tumor is a benign tumor. Cells in a malignant tumor are progressively invasive and tend to develop tumors in other healthy tissues of the body. Malignant tumors grow at multiple sites in the body and also grow repeatedly even after surgical removal. The ability of tumor cells to move from one site of the body to another for developing new tumors is known as metastasis. Malignant tumors can be classified based on the embryonic origin of the tissues from which it has been developed. If the tumor has developed in the endodermal or ectodermal tissue such as skin or epithelial linings of internal organs and glands, it is termed as carcinoma. The tumors developing from mesodermal connective tissue such as bones, fat, cartilage are known as sarcomas. Malignant tumors of blood cells, the hematopoietic cells of bone marrow are known as leukemias and lymphomas. Single cell proliferating tumors are leukemias and those which grow as tumor masses are lymphomas. Oncogenes. Oncogenes are the genes present in normal cells of the body which are required for normal functioning of the cells such as cell division and cell death. Certain changes in these gene sequences can lead to erroneous functions which can eventually cause cancer. Therefore, they are known as cancer causing genes. These genes are present in normal cells as well as some viral cells. There is a high degree of homology between the genes present in normal cells and viral cells. The copy and sequence of genes present in normal cells is known as the cellular oncogenes or proto-oncogenes and are denoted by CONC. The version of genes present in viruses is known as viral oncogenes and are denoted by VONC. Most of the oncogenes have originated from normal cellular genes. These genes carry out normal cellular functions and are called as inactivated cancer causing genes, the proto-oncogenes. When they get activated and cause cancer, they are termed as oncogenes. They can be grouped into three categories: the genes involved in induction of cellular proliferation, the genes involved in inhibition of cellular proliferation, and the genes involved in regulation of programmed cell death. Induction of cellular proliferation. The genes involved in proliferation of cells are highly regulated. The regulation mechanism checks and allows the proliferation of cells only when needed. Loss of this regulation may cause increased cellular growth. This category includes growth factor genes and growth factor receptor genes. Cellular proliferation increases because of increased expression of these genes. Genes in this category are FMS, ERBB and NEU. Inhibition of cellular proliferation. This category of genes is also known as cancer associated genes or tumor suppressor genes. They code for inhibition of excessive cellular proliferation. The normal copy of genes in normal cells is involved in the inhibition of cell division process. The cell division should be stopped as soon as the required number of cells have been produced. If there is a mutation or any other modification in these set of genes, the cell proliferation will not stop and the resultant number of cells will be very high as compared to the normal cell number. This may eventually lead to development of tumor masses. Regulation of programmed cell death. These genes are involved in induction or blocking of apoptosis. The cells in the body which are no longer required should be removed. Apoptosis is a regulated programmed process of cell death. If there is an error in the genes inducing apoptosis, the cells will survive for longer period than required, and similarly, if the genes blocking apoptosis are upregulated or highly activated, it will not allow the cells to initiate apoptosis, which eventually will lead to increased cell number and cell survival. The oncogene BCL2 falls in this category. Proto-oncogenes can be converted to oncogenes. Mutations or genetic rearrangement caused by viruses or carcinogens in the oncogenes can convert inactivated proto-oncogenes into cancer causing oncogenes. Certain cells may contain multiple copies of oncogenes. The activation of oncogenes usually occur due to translocation, physical or chemical carcinogens or viral integration. Translocation of oncogenes from one chromosomal site to another can convert proto-oncogenes to oncogenes. Burkitt's lymphoma is caused due to translocation of CMYC, a transcription factor gene, from its normal position on chromosome number 8 to chromosome number 14 near the immunoglobulin heavy chain enhancer. This leads to its increased synthesis. Physical and chemical carcinogens lead to mutations in the oncogenes and activate them to cause cancer. Single point mutation is observed in case of bladder, colon and lung carcinomas. Integration of viral genome also transforms a normal cell into a malignant cell by converting a proto-oncogene into an oncogene. Tumor antigens. Tumor antigens are grouped into two categories: tumor specific transplantation antigens and tumor associated transplantation antigens. Tumor specific transplantation antigens. Tumor specific antigens are unique to tumor cells and do not occur on normal cells in the body. They are exclusively expressed by the tumor cells. These antigens are encoded by variant forms of normal genes that have been altered by mutation. These mutational changes lead to altered cell surface proteins. Some antigens are tumor specific. For example, the antigens produced on tumor cells developed because of chemical or physical carcinogens will be different every time a new tumor is developed. Immunity developed against one tumor will not protect the animal against another tumor. However, viruses induce tumors which possess similar type of antigens. Tumour-associated transplantation antigens. Tumour-associated antigens are not unique to tumor cells. They are expressed on normal cells during fetal development, but not in the adult individuals. Tumour-associated antigens may also be proteins that are normally expressed at extremely low levels on the normal cells, but are expressed at much higher levels on tumor cells. Oncogene proteins as tumor antigens. Most of the antigens present on tumor cells are not unique to tumor cells, but are present on normal cells also. These antigens are basically the oncogene proteins. This increased expression of oncogene proteins is recognized by cells of immune system, and immune response is mounted against it. Example, NEU protein is expressed in high number on cell surface in the case of breast cancer. The immune system marks the tumor cells as different because this normal antigen is present on the cell in highly excessive number. For example, epidermal growth factor EGF binds to the receptor present on normal cells for its activation. A tumor cell may carry 100 times more the number of epidermal growth factor receptors on its surface than the normal cells. The abnormally high number of receptors on cell will definitely result in extremely high cell growth. Another tumor associated antigen is transferrin growth factor. It is designated as P97, and a cell carries 8,000 copies of the molecule, whereas a tumor cell exhibits 50,000 to 500,000 molecules per cell. Oncofetal tumor antigens. These antigens are present only on fetal cells normally during early embryonic development, when the immune system is not completely developed. If they are expressed later in life, they are considered as non-self and immune response is mounted against it. They are expressed in normal cells only in case of some cancerous growth. Mainly two antigens are found associated with cancers such as alpha fetoprotein in case of liver cancer and carcinoembryonic antigen in case of colorectal cancer. Tumors can induce potent immune responses. Tumor antigens can induce both cell mediated as well as humoral immune response. Cell mediated immune response is known to play major role in destroying the tumor cells. The tumor cells exhibit MHC on their surface. The cells present tumor antigen fragments on self MHC. These antigen fragments are recognized by the T cells. The T cells get activated and differentiate into cytotoxic T cells. The activated T cells destroy the tumor cells. Immune surveillance theory. Immune surveillance theory states that the immune system keeps a check on all the body tissues and organs and eliminates all the abnormalities wherever detected. The cells of the immune system patrol the body parts continuously to detect the kind of abnormalities occurring in the body. Cancer cells normally develop in the body which are recognized and eliminated. The cancer cells which hide their tumor antigens or impair the immune response escape from the immune cells. Tumor evasion of the immune system. Antigens on tumor cells are recognized by the cells of immune system and eliminated, but still large number of people suffer from cancer every year. This clearly indicates that tumor cells possess mechanisms to escape from the immune system. Tumor cells adopt following methods to escape immune system. Anti-tumor antibodies can enhance tumor growth. Antibodies can modulate tumor antigens. Tumor cells frequently express low levels of class 1 MHC molecules. Tumor cells may provide poor co-stimulatory signals. Anti-tumor antibodies can enhance tumor growth. The antibodies generated by tumor antigens bind to the tumor cell surface and mask them. The antigens are therefore not free for the cytotoxic T cells for initiating the tumor cell killing mechanisms. In presence of antibodies, the tumor growth increases and spreads further. Antibodies can modulate tumor antigens. Tumor specific antigens disappear from the surface of tumor cells in presence of anti-tumor antibodies and reappear after the antibodies disappear. This is known as antigenic modulation. The antibodies bind to the antigens on the tumor cell surface and form a complex. The tumor cell may shed this complex or endocytose it. In both the cases, the antigen is not visible to the cytotoxic T lymphocytes, which leads to progressive tumor growth. Tumor cells frequently express low levels of class 1 MHC molecules. The cell mediated immune response plays an important role in controlling the tumor growth. The cytotoxic T lymphocytes identify the foreign antigens through major histocompatibility complex, the MHC. These cells can be activated by the tumor antigens presented on self MHC. The tumor antigens on self MHC are recognized by T cell receptors on cytotoxic T cells. The association between MHC, tumor antigen and T cell receptor gives a signal to T cell and it gets activated. The tumor cells lower down the expression of MHC on their cell surface, which results in decreased sensitization and activation of cytotoxic T lymphocytes. This in turn increases the tumor growth. Tumor cells may provide poor co-stimulatory signals. The immune response against tumor cells is generated by activating cytotoxic T cells through MHC, tumor antigen and T cell receptor complex. The cytotoxic T cells also require co-stimulatory signals for activation. The co-stimulatory signal is initiated by the binding between B7 ligand of antigen presenting cell and CD28 on the T cell. Tumor cells do not allow efficient co-stimulatory signal, which leads to very poor immune response. Cancer immunotherapy. The immune response is generated against all the tumor cells developing inside the body, but if the response is not efficient enough to eliminate all the tumor cells, it may lead to a cancerous state. The tumor cells which become cancerous are able to impair the immune system and grow progressively. Cancer immunotherapy deals with the methods for boosting up the immune response against tumor cells. Manipulation of co-stimulatory signals can enhance immunity. The immune response against tumor cells can be enhanced to eliminate them. The co-stimulatory signal can be enhanced by transfecting the gene coding for B7 ligand in tumor cells. The B7 ligand produced by the tumor cells will provide enhanced co-stimulatory signal to T cells and activate it. Enhancement of antigen presenting cell activity can modulate tumor immunity. Antigen presenting cells can activate T helper cells and cytotoxic T cells against tumor antigens. Growth factors for activating the antigen presenting cells are transfected inside the tumor cells and infused into the patient. The genetically engineered tumor cells enhance the differentiation and activity of antigen presenting cells. These antigen presenting cells further activate the T helper cells and cytotoxic T cells. The activated T cell repertoire destroys the tumor cells. Cytokine therapy can augment immune responses to tumors. Cytokines can increase the immune response against tumor cells by activating and differentiating various cells of immune system. Cytokines like interferon alpha, beta and gamma, interleukin 1, interleukin 2, interleukin 4, interleukin 5 and interleukin 12, granulocyte monocyte colony stimulating factor and tumor necrosis factor are known to influence the immune response. They are cloned and produced at large scale for the clinical applications. This option of cancer immunotherapy is still at its research level. Interferons are administered to cancer patients by injections. Many cases have shown promising results with considerable tumor regression. Interferons increase the expression of MHC, which further enables the activation of cytotoxic T cells. Tumor necrosis factor is an anti-tumor cytokine. It is involved in killing of tumor cells and reducing the cell proliferation. Monoclonal antibodies are effective in treating some tumors. Monoclonal antibodies have been applied in some cases of human B cell lymphomas. Antibodies are raised against the idiotype of the antibody present on the cancerous B cells. All the cancerous B cells will possess antibodies having same idiotype. Therefore, the monoclonal antibodies will bind to all the B cells and eliminate it. Immunotoxins are the toxins attached to the monoclonal antibodies. They are also known as guided missiles because they attach to only the specific antigens against which it is directed and releases the toxin for the destruction of tumor cells. Cancer is a group of diseases which has become a major cause of deaths in last few years. The active research going on in the field of tumor immunology has enabled the scientists to develop new treatment methods. The advances in cancer immunotherapy will definitely provide solutions to the present problems.